Welcome back from the break. So we got some questions. There was a problem with the webinar system, and we had some delay in getting the questions. So before we go to the next presentation, we will answer some questions regarding the previous presentation. So I will first of all I will say that the webinar is being recorded. We plan to put this recording publicly available fairly soon, as soon as we can. We plan to share the webinar slides also. And then there are some questions related to growth factors and the studies, the preclinical studies. And maybe we start, Mart, could you briefly make a few comments on the role of growth factors in the normal brain, healthy brain?
Yeah, thank you. So this has been studied quite extensively in animals using modern transgenic methods. For example, deleting the gene for CDNF or its receptor, or nurturing or its receptor, and we have recently also deleted the gene of the CDNF. So in the case of the absence of the CDNF, there is a functional impairment in dopamine neurons in the midbrain. We do not see their loss, but we see that they do not function anymore normally. Then what we see also in these animals is that they have actually lost dopaminergic neurons in the gastrointestinal tract. And these animals develop a condition which is very similar to patients, constipation. So if we delete the CDNF receptor RET, we actually see age-dependent loss of dopaminergic neurons, although a little bit more modest than in Parkinson's disease, but still. So we can tell that growth factors are important for the survival, maintenance, and functioning of growth factors in the normal brain. In many cases, it has been also shown, and there is a beautiful study now on the CDNF-related growth factor month, that the levels of these growth factor are dramatically reduced in aging. So they obviously also play a role in aging.
Thank you. We maybe have a time for a couple of more questions. So I'll briefly mention that the MPTP rhesus monkey results have not been published yet. There is a manuscript that is in fairly late stages, and hopefully this will be published hopefully next year. It's been a long process, but of course the study is important to get publicly evaluated and available. Then there's a couple of more questions related to the CDNF mechanism of action. How does CDNF affect proteostasis? And have there been any studies that show improvement of dopamine neuron function when doing more than 6-month treatment after injury? And are there some studies available where CDNF therapy is compared to, for example, stem cell transplant effects?
So
just briefly, Martin. We don't need to go to details.
Yeah, thank you so much. So actually the longest study on the effect of CDNF in vivo has been this current clinical trial. We have treated animals until 6 months, and we don't have data on the longer effects. So on the mode of action, I think there are several papers now published independently from our laboratory that CDNF is actually reducing the ER stress, reducing the signaling by UPR, unfolded protein response pathways. And this is also very nicely seen in the CDNF knockout or in the MANF knockout. These animals which lack CDNF have in several tissues chronically activated signaling of unfolded protein response pathways. So I think we are now very close in reporting also about the receptors. Our paper on the month receptor, on the cell surface receptors, was just last week accepted in eScience, and we are going to publish about the month ER receptor also in the coming month.
Good, thank you very much. I think now we have to move on with the program. These were good questions and very good answers. So next, Magnus Sjögren. Magnus, could you turn on your camera? And Rebecca has the presentation ready. So we will now move on and present first the safety and then the secondary and exploratory endpoints of the clinical study. Please, Magnus.
Yes, thank you, Henry. Yes, Magnus Sjögren, Chief Medical Officer of Herantis Pharma. And I'm going to walk you through a little bit on the first human clinical trial with CDNF in Parkinson's disease, the 12-month data, and start out with presenting the design of the study. So please, next slide. So this clinical trial enrolled 17 individuals with Parkinson's disease. The primary endpoint was safety and tolerability, while we also, as secondary endpoints, studied efficacy of CDNF in these individuals. It was a clinical trial conducted at 3 clinical sites, Karolinska University Hospital in Stockholm, Sweden, Skåne University Hospital in Lund, Sweden, Helsinki University Hospital in Finland, and also 2 PET centers. And PET is positron emission tomography technology to visualize dopaminergic neuronal activity in the brain. And these 2 sites were Karolinska Institute PET Center in Sweden and Turku PET Center in Finland. Next slide, please. So this is an overview of the clinical trial design. On top of this slide, you can see the study was divided into 3 parts, while 2 were with active treatment. And that is to the upper left, the main study, which was a 6 months part, which included monthly infusions of either placebo or CDNF. And an extension study for 6 months, continuing with the CDNF, and then a follow-up study which is currently ongoing and will continue for 4 years in total. So back to the first main study part, 6 individuals were randomized to placebo, and the design was double-blind. Placebo controlled compared to mid-dose CDNF, which 6 individuals received, and high dose, which 5 individuals received. And then they were, the placebo exposed individuals were re-randomized to receive mid-dose of CDNF or high dose of CDNF. So the extension study for 6 months, all enrolled individuals received active treatment and continuing with monthly infusions of CDNF. The safety part does not include any active treatment, but safety assessments alone. So in the lower part of this slide, we have an overview of some of the clinical characteristics, and you can see the age group was similar in the placebo as in the mid-dose group, which is the 3rd column of this table. Versus the high dose, which is the last column of where the individuals were slightly younger, on an average 58 years of age. And the disease duration was slightly more than 10 years in each of these 3 groups. And you can also see, you can spot perhaps in the headings of the different columns here that for CDNF, we had a titration scheme starting with low and then moving on towards mid, ending up in either mid or a high dose in the 2 active treatment groups. Also, the off symptoms on an average were slightly more than 4 hours per day at the baseline. Next slide, please. With regard to clinical trial procedures, altogether there were 26 visits over a period of 16 months. And that included, of course, the 12 treatment infusions once monthly. And there were many brain scans and other scans taken for before and for planning of the surgical procedure and for safety assessments. And then we had the neurosurgical implantation of the drug delivery device. And you can see both a drawing on top right corner here of this slide, depicting the drug delivery device as it is supposed to be when implanted on top of the skull of an individual. And my colleague from Renishaw, Jim Baker, will very soon present a video on the implantation procedure and the device. And on the lower right part of this slide, you can see a photograph of one of the individuals that has a port implanted just behind the right ear, as it is on this depicted on this slide here. So moving back to the procedures, there were a number of off-medication sessions with overnight stays, travel to the PET center, and data collected and delivered to the research nurse and the investigator to assess safety of the individuals as well as efficacy. And the individuals who were enrolled and their spouses, they were instructed on a daily maintenance procedure to ensure that the skin around the port was clean. So this was clearly a very intense clinical trial for the patients and the clinical staff that worked with this trial. So once again, I'm extending our gratitude to everyone who has been involved in this trial, the patients especially, and their spouses for all their work on this, and the clinical staff who have done an excellent work with ensuring the proceedings of the trial. So we'll move into the next slide, where my colleague in Renishaw Neurosolutions will present the drug delivery system and the device. Please go ahead.
Thank you, Magnus. So you can see here a 3-tube configuration to the catheters, an inner and outer guide tube and a catheter. The 3 tubes fit together to form a kind of recessed step at the end, a unique feature to the Renishaw drug delivery device. You can see it there. So with respect to the implantation, the surgeon first forms a hole in the skull into which the outer guide tube is pushed using the stereotactic brain surgery. You'll just see that going in a minute. Yes, so the hub of that outer guide tube fits into the hole, and then the inner guide tube is inserted. And this here is a stylet, which can be used by the surgeon if they want to, and it's visible in medical imaging, so they can verify the position at which the catheter will be. So then the stylet's removed, and the actual catheter itself is inserted down that 2-tube configuration. And this forms the recess step at the end. So you can have up to 4 catheters implanted, and they come together into a port which comes through the skin, and the port is mounted behind the patient's ear. And to that port you can see a giving set that can be attached in an outpatient situation when an infusion is required. The infusate is pushed down using syringe pumps and gently pushes out the infusate, which, due to the nature of that recess step, does not track up the outside of the catheter, but pushes the infusate out into the target. And you can see there, bacterial filters on the lines for extra safety.
Thank you, Jim. So we continue with the presentation of the safety results from this CDNF first-in-human clinical trial in Parkinson's disease, a 12-month trial. Next slide, please. So there were numerous safety assessments done in this clinical trial. So at every visit, the patient and their spouses were asked about any potential new adverse event that had occurred in between the visits. And it may be related or not, any event was recorded. There were also a range of laboratory tests done on blood and urine that had been withdrawn. That included also tests for anti-CDNF antibodies, that is, antibodies toward the active medication being CDNF, and that may indicate a reaction towards the drug that could potentially hamper its effect. There were also a lot of physical examinations done both at screening before treatment start and every 6 months, which included also a neurological examination and electrocardiogram. And every 3 months, there was a brain magnetic resonance imaging scan done for safety, but also to ensure distribution of CDNF. In addition, there were the patients completed a range of questionnaires, for example, covering aspects of cognition, mood, and impulsive-compulsive behavior, which are domains, behavioral domains that are known to change with Parkinson's disease, and these were done for safety. And in any case of any event that was deemed medically relevant, there were additional visits to the investigator to follow up on these. Next slide, please. So this is the observed adverse event profile. So for all adverse events reported, whether or not related to the drug or to the treatment, this is the overall picture of any adverse event. And as you can see on this pie chart to the left, the majority of the adverse events were in the so-called nervous system domain or disorders domain. And I'll give some examples very soon on what that means. And you can see the other domains. The next one was general disorders and administration site conditions, a type of adverse events, followed by psychiatric disorders, injury, poisoning, procedure complications, infections and infestations, gastrointestinal disorders, musculoskeletal and connective tissue disorders, skin and subcutaneous tissue disorders, and something related to adverse events related to investigations, and then adverse events classified as others. Next slide, please. So as promised, these were the most commonly reported drug-related adverse events. So there were 68 adverse events that were judged as drug-related, that is related to CDNF, over this 12-month treatment period. And for central nervous system disorder class, these were frequently dyskinesias, indicating a worsening of the disease when or if present. Headache, and another example was small air bubbles in the brain, which were judged as non-significant and non-symptomatic. Jim Baker, who presented on the drug device just a couple of minutes ago, will come back to this and describe this in a little bit more detail. And then we had psychiatric disorders. That examples of that was impulse control disorder. General disorder was feeling of cold, fever, and tiredness. Gastrointestinal disorder examples were nausea, and other disorders examples were weight loss. So next slide, please. As for serious adverse events, whether or not related to the drug or the drug delivery system or the surgical implantation of the drug delivery device, and the definition of serious adverse event is an adverse event that requires either hospitalization or that may be life-threatening. So altogether, there were 9 serious adverse events reported over the 12 months study period, and 1 occurred prior to surgery. That was a purulent sinusitis. And since it was before initiation of any procedures related to the device, or the surgery, or the CDNF infusions, this was obviously not related to treatment. There were 5 serious adverse events that occurred after surgery and prior to treatment start, that is prior to infusions of CDNF, 2 confusion reactions, and then there was an infection of the skin around the port, skin tissue decay, or as it may be called, necrosis around the port, and 1 case of wrist fracture after a fall. Then, as the infusions of CDNF started, there were an additional 3 serious adverse events and 2 cases of brain abscesses and 1 general infection not otherwise specified. So next slide, please. So with regard to these serious adverse events that may be related then to CDNF, there were 2 cases of brain abscess and 1 that followed surgery, skin tissue decay, and all of these, including the previous ones, obviously induced internal conferences between Venisho and Herantis and investigators, where we developed risk mitigation actions to ensure the safety and continuation of the trial. So these risk mitigation approaches were made to improve the surgical procedure, infusion procedure, device maintenance procedure, and there was also additional training of the investigators, the surgeons, and the neurologists. And after these risk mitigation improvements that were made, there were 87 infusions conducted without any infusion procedure related or other procedure related adverse event. Next slide, please. So this is the summary of the 12-month first-in-human CDNF clinical trial safety data. So there were CDNF was regarded as safe and well tolerated, and the majority of the reported drug-related adverse events were mild, and all patients have recovered. In addition, there was a similar safety profile in the main and the extension study, and less reported adverse events in the extension part. And there were no clear differences in safety profile between the treatment groups. So between placebo, mid, and high dose, there were no clear differences between these 3 groups with regard to safety, and there were no dose-limiting toxic effects of the drug. So to conclude, CDNF, as based on this first-in-human trial in 17 individuals with Parkinson's disease, was confirmed to be safe and well tolerated, drug safety established, and this enables us to move forward with CDNF for clinical development, further clinical development. And with regard to the drug-device combination, these safety improvements that were made enabled to improve safety moving on to the trial. So this is my last slide, and I thank you for your attention, and I will now give the word to Professor Per Svenningsson, who was the primary investigator of this